Hi everyone, welcome to the cell biology module again. So now in this chapter 5, we are going to learn the structure of DNA, and we also are going to learn the concept of this protein synthesis. So first of all, I will start with this nucleic acid. So nucleic acid is in fact is a polymer, is a type of macromolecule as well, same as carbohydrate, lipid, and also protein. So since they are macromolecule or a polymer, surely they have their monomer. They are formed by many same subunit. So the subunit of nucleic acid is called nucleotide. So nucleic acid, in fact, they have two types of nucleic acid. One is called deoxyribonucleic acid. The other one is called ribonucleic acid. Deoxyribonucleic acid is the DNA that we commonly say. What is the function of DNA? In general, I can say the function of DNA is to control the activity of cell. Or I can say DNA carries genetic information of a cell or an organism. Well, ribonucleic acid we can also call as RNA. RNA, the function of RNA is involved in protein synthesis. So both DNA and RNA, they belongs to the macromolecule nucleic acid, and both DNA and RNA, they are polymer formed by monomer or formed by subunit called nucleotide. How the nucleotide looks like? Each nucleotide is consists of three components: a pentose sugar, or we can call them as 5-carbon sugar, a phosphate group, and also a nitrogenous base. This is the structure of a nucleotide. So nucleotide consists of a pentose sugar, or we call it as 5-carbon sugar, linked to a nitrogenous base. The nitrogenous base is containing nitrogen element. That's why we call them as nitrogenous base. And the other side of the pentose sugar is linked to a phosphate group. The phosphate group itself is carrying two negatively charged. That's why DNA and RNA, they are negatively charged. And this pentose sugar is having 5 carbon. If you look at the picture here, it's showing the position of the carbon. This is carbon 1. Carbon 1 is the one that linking to the nitrogenous base. This is carbon 2. This is carbon 3. This is carbon 4. While carbon 5 is at the between, the bond between the phosphate group and also the pentose sugar. This is just a picture illustration of the structure of this nucleotide. In fact, the structure, it looks like this in reality. This is a nitrogenous base. It has nitrogen element in its structure. And this is the pentose sugar, while this is the phosphate group. Since the nucleotide of DNA and RNA, since both DNA and RNA, they are formed by nucleotide as a monomer. So you may be asked, is there any differences between the nucleotide in DNA and also the nucleotide in RNA? The answer is yes. There are two main differences between the nucleotide in DNA and the nucleotide in RNA. So the first difference is in the 5-carbon sugar, which is a pentose sugar. If you look at the picture here, by comparing the nucleotide in RNA and this is a nucleotide in DNA, you may be noticed that at carbon number 2, which is this position, in DNA, the 5-carbon sugar at carbon number 2, it is H hydrogen that attached to the carbon 2 in the 5-carbon sugar. While in RNA, at carbon number 2, the one that attached to the 5-carbon sugar at carbon number 2 is OH. But in DNA, it is H only. So that's why in DNA, this 5-carbon sugar, also known as deoxyribose sugar. While in RNA, this sugar is also known as ribose sugar, or we call as ribose. So that's why, so the difference between the 5-carbon sugar in DNA and also RNA is, in DNA, the 5-carbon sugar at carbon number 2 is H only, without oxygen, without O. So that's why where the term deoxyribose sugar come from. The term D mean remove, oxy mean oxygen. So which mean the oxygen have been removed in the 5-carbon sugar in DNA. That's why we call it as deoxyribose sugar. So in summary, which means the first difference between the DNA and RNA in terms of their monomer is, in RNA it is ribose sugar, while in DNA it is deoxyribose sugar. The second difference is the nitrogenous base. So we have 5 types of nitrogenous bases. There are adenine, guanine, cytosine, thymine, and uracil. So out of these, adenine and guanine is formed by 2 carbon rings, so we call them as purine. While cytosine, thymine, and uracil, you may be noticed that all of them formed by 1 carbon ring only, so they are known as pyrimidine. However, out of these five of them, not all of them are found in both DNA and RNA. In DNA, it has only adenine, guanine, cytosine, and thymine. While in RNA, it has only adenine, guanine, cytosine, and uracil, which means in DNA there is no uracil. In RNA there is no thymine. So for them to form the nucleotide, the DNA will choose one out of the 4 components of the nitrogenous base, which means in DNA they will choose one nitrogenous base out of adenine, guanine, cytosine, or thymine, one of them, to form the nucleotide. While for the RNA, same thing, they will choose one of the nucleotide, one of the nitrogenous base out of the 4 choices, which is adenine, guanine, cytosine, and uracil, one out of them to form their nucleotide. So in summary, the second difference is in DNA, it had the nitrogenous base of DNA has only adenine, guanine, cytosine, and thymine. There is no uracil in DNA. While in RNA, the nitrogenous base is adenine, guanine, cytosine, and uracil. There is no thymine in RNA. These are the 2 major differences between DNA and RNA in terms of their nucleotide. So when 2 nucleotides they join together, they will form polynucleotide. How 2 nucleotides can be joined together? The 2 nucleotides can be joined together through formation of a bond called phosphodiester bond. This phosphodiester bond link between the 5 carbon sugar to the phosphate group. From here, they will form a strong covalent bond, which is a phosphodiester bond. Now, let's learn about the structure of DNA. The structure of DNA is according to the Watson and Crick model, the 2 scientists that discover the structure of DNA. First of all, we know that the DNA is formed by the nucleotide. The nucleotide of DNA can also call as deoxyribonucleotide. The deoxyribonucleotide consists of a 5 carbon sugar, or you can call them as deoxyribose sugar, 1 nitrogenous base, and a phosphate group. Two nucleotides can be linked together through formation of phosphodiester bond. So the sugar and phosphate group will form the backbone of DNA. What is the meaning of backbone? As we know, DNA is double helix, so you always see the DNA is twisted in a helical form. The twisted helical form, in fact, is showing the sugar and the phosphate, while the nitrogenous base is projected to form at the middle, the center of the double helix. So the sugar and phosphate is forming the part where the DNA can be twisted, so we call them as sugar-phosphate backbone. When a lot of nucleotides are linked together, then they will form a polynucleotide. This is a polynucleotide formed by a lot of nucleotides. So you will be noticed that they contain the 5 carbon sugar, the phosphate group, and also the nitrogenous base. Each nucleotide are linked together through formation of phosphodiester bond. While the other polynucleotide will be linked to another one. So 2 polynucleotides will be linked together. From here, it form a double-stranded. That's why we always say DNA is a double-stranded, because it is formed by 2 polynucleotides which link together. How the 2 polynucleotides can be linked together? Through the nitrogenous base at the center. So the nitrogenous base will be linked together through the formation of hydrogen bonding. Adenine always pair to thymine. Guanine always pair with cytosine. Adenine will form 2 hydrogen bonds with thymine, while guanine will form 3 hydrogen bonds with cytosine. So from here, 2 polynucleotide strands can be linked together, and here it forms double-stranded of DNA. And you may be noticed that the other polynucleotide, they are in inverted position. So if you look at the structure here, the one that highlighted in black, they are the sugar phosphate backbone. And if you look at the position of this carbon, so this is a 5-carbon sugar. If you look at my arrow here, so my arrow is pointing to carbon 1 in this 5-carbon sugar. This is carbon 2. This is carbon 3, this is carbon 4, and this is carbon 5. So if you look at this picture here, which carbon is at the top position? Is this carbon, which carbon 5? So that's why this end is also known as 5' end. And if you look at this picture here, this is the lowest nucleotide. And if you look at my arrow pointing to the carbon number, this is carbon 1 in this 5-carbon sugar. This is carbon 2. This is carbon 3. This is carbon 4. This is carbon 5. So in this nucleotide, since it is at the lowest position at the bottom, so which carbon is at lowest position and outermost? This carbon, which is carbon 3. So that's why this end also known as 3' end. So that's why DNA has a position or direction of 5' to 3'. The 5' and the 3' is indicating the carbon numbering. So at this position, the top carbon is carbon 5. So that's why known as 5' end. While at this position, the lowest and outermost carbon is carbon 3. That's why also known as 3' end. Same thing to this strand, which is in the inverted position. Where's carbon 1? Carbon 1 is at here, the one that linking to nitrogenous base. This is carbon 2, this is carbon 3, this is carbon 4, and this is carbon 5. So which carbon is at the top position? This one, carbon 3. So this side is known as 3' end. And in here, where is the carbon? Where is the carbon at the lowest position? This is carbon 1, this is carbon 2, this is carbon 3, this is carbon 4, and this is carbon 5. So the lowest position is carbon 5. So that's why this end is known as 5' end. This end is known as 3' end. So you may be noticed that one of the polynucleotide is in upright position while the other one is in inverted position. So we say they are arranged aligned in anti-parallel. So in DNA is double-stranded and twisted into helical structure. The polynucleotide, they are aligned in anti-parallel to one another. How about in RNA? RNA is formed by a lot of ribonucleotide. They link together. However, RNA is only single strand, not double-stranded. And you may be noticed that in RNA, there is no thymine. The thymine has been substituted into uracil. However, the structure of RNA we'll discuss later. So the structure of this DNA is known as Watson and Crick model, discovered by two scientists that won Nobel Prize in 1963. Where do we find the DNA? In fact, DNA is found in chromosome. So if you remember in chapter 4, I did tell you. So humans' DNA is very long. How can we pack this DNA into very smaller structure of cell? In fact, the DNA will coil around a type of protein in human, which is called histone protein. When DNA coil around histone protein, they'll further condense into a structure called chromatin. Chromatin will be further condensed into chromosome. So DNA is found in the chromosome. So this chromosome is found in the nucleus. So always we say the function of DNA, or we say the function of chromosome, or we say the function of nucleus. Actually, all of them, they are the same. The function of nucleus is same as the function of chromosome, is same as the function of DNA, which is control activity of cell or carries genetic information of an organism or a cell. So this is a structure of DNA which have been twisted into double helix structure. So the sugar phosphate backbone consists of the 5-carbon sugar and the phosphate group, while the nitrogenous base is projected at the center. Here it forms a genetic code. So when we talk about the genetic code, it is always referring to the sequence of nitrogenous base in the DNA. How about RNA? RNA, also known as ribonucleic acid. The monomer or the subunit of RNA is known as ribonucleotide. All of the RNA, they are single-stranded instead of double-strand. And RNA has 4 types of this nitrogenous base: adenine, uracil, cytosine, and guanine. Out of 4, one of them will be chosen to form the nucleotide. And in the sugar, the 5-carbon sugar in the RNA is ribose sugar instead of deoxyribose sugar. So what is the function of RNA? The function of RNA is involved in protein synthesis. So in fact, we have 3 types of RNA. DNA, it has only 1 type, but RNA, it has 3 types. 3 types of RNA are found in all the cells. The first one is messenger RNA, or we call them as mRNA. In general, messenger RNA, it carries genetic information from DNA to the cytoplasm. What is the purpose? The purpose is to synthesize protein. While the other type of RNA is called transfer RNA, or we call it as tRNA. tRNA or transfer RNA is to carry amino acid, and this amino acid will be delivered to the mRNA. Later on, they can be used to form protein or polypeptide. The other type of RNA is called rRNA or ribosomal RNA. This type of ribosomal RNA is found in the ribosome. It is just something like they are very shy, so they'll be hiding inside the ribosome. So they're found in the ribosome. The function of this rRNA, or you can say the function of ribosome, is to hold mRNA and tRNA together so that they can work together to synthesize protein. Now let's go into a little bit detail. For this mRNA or messenger RNA, it is in single-stranded. However, in the mRNA, for every 3 bases in mRNA, we call them as codon. So what is codon? Codon is every 3 bases in mRNA, we call them codon, or we say as triplet base in the mRNA, we will call them codon. The term codon can only be used in mRNA. You cannot use the term codon for DNA, for tRNA, also for RNA. Cannot. Only the triplet base in mRNA, we can use the term codon. While for tRNA, the function is to transfer amino acid. Although this DNA looks like in this shape, however, it is still single strand. If you look carefully, the position from here, 3' is just folded into this type of structure at the end to form 5'. So in the end, it is just still single strand, but have been folded into this called cloverleaf shape. The function of tRNA is to transfer amino acid. Where you're going to transfer the amino acid is at this position called amino acid attachment site at the 3' end. The amino acid will be attached at here. And at the other end, the triplet base in the tRNA is called anticodon. This anticodon formed by 3 bases in tRNA and is only at this position only, which means at the other position of the triplet base in tRNA, you cannot call them as anticodon. The term anticodon is used in tRNA only, and only at this position. The rest position in the tRNA, you cannot call them as anticodon. What is the function of anticodon? The function of anticodon is used to code the codons in mRNA so that they can be used to synthesize polypeptide. While for ribosomal RNA or rRNA, actually they look like that. But once they are synthesized in nucleus, they will be packed with a protein later on to form a functional ribosome. So in fact, inside ribosome, it consists of rRNA and protein. Ribosome has small subunit and large subunit. What is the function? The small subunit is used to hold mRNA, while the large subunit is used to hold tRNA, so that the tRNA can use its anticodon to read, or we say to code the codon of mRNA so that they can synthesize protein. This is differences between DNA and RNA. It is very important for you to know these differences between DNA and RNA. First of all, in terms of sugar, in DNA, the 5-carbon sugar is deoxyribose sugar, while in RNA is ribose sugar. So in DNA, the sugar at carbon number 2 is only H without O. So that's why we call it as deoxy. In terms of the nitrogenous bases, in DNA it has adenine, thymine, cytosine, and guanine, while RNA has only adenine, uracil, cytosine, and guanine. DNA is double-stranded. DNA is only single-strand. While DNA forms helical structure, RNA does not. We have only one type of DNA, but RNA we have three types: mRNA, tRNA, and RNA. The function of DNA is to carry genetic information, while the function of RNA is involved in protein synthesis. DNA is longer than RNA because DNA carries genetic information of a cell or an organism, while RNA is only works to synthesize protein, so it's much shorter than DNA. DNA is found in the nucleus, while RNA is found in nucleus and cytoplasm. But remember, this is only true for eukaryotic cell, which has nucleus. For prokaryotic cells, such as bacteria, their DNA and their RNA are all found in cytoplasm because prokaryotic cells do not have nucleus. This is the structure of DNA and RNA. Now, we come to another part that is talking about the history. How do scientists they are able to discover that DNA is the genetic material or heredity material? So in fact, long time ago, actually the scientists they do not know that what is the heredity material or what is the genetic material that determine the characteristic of the cell. So by this, we have set a few scientists, they do a lot of experiment from there, they confirm that in fact DNA is the heredity material, because during that time there are a lot of argument. Some of the scientists they thought that protein is the heredity material, not DNA. So that's why a few scientists like Griffith, Avery, Hershey and Chase, they conduct a few experiments. From the experiment, they confirm that DNA is the heredity material, not other macromolecule. You can click this YouTube link for detailed explanation, the animation to explain about this experiment. After understanding how scientists discover that the DNA is the genetic material, here let's look at another concept called DNA replication. What is the meaning of DNA replication? DNA replication simply means how DNA molecule it can turn into two from originally it has one molecule. So when DNA molecule is duplicating, this is called DNA replication. So initially there are three theories explaining how the DNA replicate. However, later on, Messelson and Storr has used experiment to prove that the method of DNA replication is by semi-conservative replication instead of the other two theory. So if you look at this figure, it explains the three model of DNA replication. In the conservative replication, if you look at this picture, here is showing the original DNA molecule, or we can call them as a parental strand. So in conservative replication, when the parental DNA forms new DNA, you will be noticed that two new DNA molecules are formed. Out of the two new DNA molecules, one of them, the one with blue color, are totally formed by newly synthesized strand, while the other DNA molecule formed consist of the parental original strand, which is the yellow color, same as the original one. In the other one, the semi-conservative replication. In semi-conservative replication, it stated that when the daughter DNA form, when the new DNA molecule form, the new DNA molecule form consists of one newly synthesized strand, which is the one in blue color, and one parental strand, which is the one in yellow color. So the new DNA molecule form consists of one original strand and one newly synthesized strand. That's why it is called semi-conservative replication. While in dispersive replication, it mentioning that the parental strand is broken into pieces and is combined with the newly synthesized fragment. So the new DNA molecule form consists of parental fragment and newly synthesized fragment. That's why it is called dispersive replication. However, the study of Messelson and Storr proven that the conservative and this dispersive replication is wrong. The correct one is a semi-conservative replication. So this diagram also explaining the semi-conservative model. So as you can see, this is a parental DNA molecule, while these two are the newly synthesized DNA molecule. So the parental DNA is having this nitrogenous base sequence. So during semi-conservative replication, the parental strand will separate. The parental strand will act as a template. So as you can see here, this is a strand 1, which is originally from here, while this is a strand 2, which is originally from here. So during semi-conservative replication, both strand 1 and strand 2, which is a parental strand, they will separate as template. So the new DNA molecule will form based on the nitrogenous base sequence in the parental strand. So from here, the nitrogenous base sequence can be linked together. From here, it forms newly synthesized strand. So you may be noticed that the new DNA molecule form consists of one parental strand and one newly synthesized strand. And please take note that on the nitrogenous base sequence, the nitrogenous base sequence of the new DNA molecule form is exactly identical 100% as the original one. So this is to ensure that the new DNA molecule form is 100% identical to the parental strand. So during semi-conservative replication, we say that each of the parental strand they serve as template for synthesis of a daughter strand. So the new DNA molecule form consists of one original strand and one newly synthesized strand. For DNA to undergo replication by synthesizing more DNA molecule, it involves a few processes. So here actually it involves three main enzymes. The first enzyme is called helicase enzyme. Helicase enzyme is used to unwind the double-stranded DNA. So once the double-stranded DNA is unwind, now the bases are exposed. So these exposed bases will be added with new nucleotide. So to add new nucleotide, you will need second enzyme, which is called DNA polymerase. DNA polymerase enzyme will help to add nucleotide to the parental strand through complementary base pairing. So by here, the nucleotide will be added one by one to the parental strand so that it can synthesize new DNA strand. Well, after added with new nucleotide, you will need to have third enzyme called DNA ligase. DNA ligase will link the adjacent nucleotide together so that this nucleotide can be linked together to form a polynucleotide strand. So from here, the parental strand and newly synthesized strand will be wrapped around to form a dihelical structure again. From here, a new DNA can be synthesized. We have already learned how the scientists discover DNA is a genetic material, and we also know that DNA replicates by using semi-conservative model. So the next question will be, we always say DNA control activity of cell, or we say DNA carry genetic information of an organism or a cell. So the question will be, how can DNA carry genetic information, or we say DNA control activity of cell? In fact, DNA control activity of cell through synthesizing protein. The protein synthesized by DNA will be used to carry out many activity, many function in the cell. So in fact, DNA control activity of cell through synthesizing protein. So DNA consists of nucleotide. The DNA undergo replication is from here it can form many DNA molecule. When a DNA synthesizing mRNA, this process is called transcription. Then from here, the mRNA will be used to synthesize protein. This process is called translation. So from here, DNA synthesizing protein. This protein will be used to control activity of cell, to carry out many functions so that to determine what are the activity the cell can be done. So this is how DNA control activity of cell by synthesizing protein. But how can DNA synthesize protein? Via synthesis of this RNA. So when DNA synthesizing RNA is called transcription. When the RNA is used to synthesize protein, this process is called translation. So this is a DNA. So in the DNA, the single strand of DNA will be used to synthesize mRNA. This process is called transcription. Take note that only one of the strand of DNA can be used to synthesize mRNA, which is the DNA in the direction of 3' to 5'. The DNA in the direction of 3' to 5' is known as template strand. It is used to synthesize mRNA, while the other counter strand, the DNA molecule in the direction of 5' to 3' will not be used to synthesize mRNA. So the mRNA synthesized will be in the direction of 5' to 3'. So the mRNA form will be used to synthesize protein with the aid of ribosome and tRNA. And take note that in mRNA, for every 3 bases in mRNA is known as codon. So 3 bases in mRNA, or we say 1 codon in mRNA, is used to determine an amino acid. What is transcription? Transcription is the process by which a molecule of DNA is copied into a complementary strand of RNA, which is known as mRNA. So from here, mRNA will be synthesized using the nitrogenous base sequence in DNA, and then later on this mRNA will move to cytoplasm to undergo translation to synthesis protein. So translation is known as a process where genetic information transferred to mRNA form DNA and is translated by ribosome to amino acid sequence on polypeptide and with the assistance of tRNA. So from here, a polypeptide or protein will be synthesized. This protein later on it can be converted into enzyme, converted into hormone, converted into antibody, or converted into signaling molecule. From here, it carry out the activity of a cell. This is a genetic code table. So based on the genetic code table, it can know which amino acid to be synthesized. Bear in mind that the genetic code table is reflecting the base sequence in mRNA only. So all the sequence here in this genetic code table is representing the codon of mRNA, not DNA, also not tRNA. This is the start codon, and these are the stop codon to determining the process of translation. How can the process of translation increase to increase the rate of translation? But actually we can done it by having polyribosome, which meaning that by having many ribosome to read the sequence of mRNA at the same time. So from here, the process, the rate to synthesis polypeptide can be increased. So in conclusion, we can say that transcription is a process, the DNA synthesizing mRNA. So this mRNA form, it may be undergoing a process of RNA processing, but this is only limited to eukaryotic cell. When the mRNA is formed